Hi everyone, I am Abhimanyu, and my presentation is about how to publish a paper without lab experiment by using single cell RNA sequencing data. Well, today is about a very practical question: how do we publish a solid paper without running new wet lab experiments? Well, the short answer is you still need novelty, just not the new benchmark. We will use public single cell RNA-seq as the backbone and focus on what reviewers actually demand. For example, clean design, donor aware statistics, replication across cohorts, and reproducibility. By the end of this seminar, you should have an end to end workflow and a figured plan you can execute as a real paper. Well, let me quickly introduce about myself. So I am Abhimanyu Thakur, and I did my B Pharm from West Bengal University of Technology, followed by M Pharm from BIT Mr. Ranchi. Then I obtained my PhD in neuroscience, working on glioma biology from City University of Hong Kong. Then I did my postdoctoral fellowships from University of Chicago and Harvard Medical Schools. Over the year, I faced several renowned awards, including Young Investigator from American Association for Cancer Research and Korean Cancer Association. And also I received Global Young Scientist Award from Nanyang Technological University, Singapore. And with the knowledge as well as the experience which I accrued, I could even publish some of the important papers in Nature Communication, Nature Microbiology, Science Advances, and Journal of Extracellular Research, as well as many more. So with that, let's define the core idea in one slide, what we exactly need for publishing a good paper by just using public database. So the core idea is you can publish without new benchmark if you deliver a new insight, robust analysis, and strong validation. The phrase I want you to remember is robustness is the wet lab equivalent, and the batch and the confounding control, replication, and reproducibility take the place of we did a new experiment. For example, public single cell RNA-seq is now big enough that you can tell multi-cohort, multi-technology stories instead of single data anecdotes. So what counts as a novel if we are not generating a new data? People often wonder. Well, the reviewers don't require wet lab for novelty. They require a non-obvious contribution, and four common novelty lanes work well with public single cell RNA-seq. For example, new cell subtype state definition with convincing markers and functional interpretation, disease relevant program, then cross-cohort meta-analysis that resolves disagreements between studies and proves reproducibility. We also need method or pipeline or the tools that improve annotations, integrations, inference, or a focused high confidence. So overall, there is a cascadal process that follows, like ligand to receptor, receptor to target, and that axis that generates the testable hypothesis. And the key point is the novelty is not a PTU map. It is a definable claim plus evidence. So now, given those novelty, this lens, what do journals still expect you to show, which can be published? So we will focus on what journals expect. Even without wet lab, journal standards stay rigorous. You need a clear biological question and why it matters. For example, replication across data sets, controls for batch effects, doublets, ambient RNA, donor level confounders, and validation beyond visualization. For example, differential expressed replications or even reference mapping, orthogonal modalities, and ideally outcomes. And reproducibility is non-negotiable. For example, code, environment, provenance, or even rerunnable figures, all these are needed. If you can't rerun it, reverse treat it like it did happen, and it cannot be published. So the first practical step is choosing a question that is publishable from public data. So here is a very good example. So for publishing, you need a good questions. The good questions are shaped like which cell type drive phenotype XYZ across cohorts, or what immune stromal crosstalk predicts progression or response, and another strand angle in concept. Why do you studies this? Why do you studies this disagree? And the transition and the classic is, can we derive a cell state signature that generalizes to bulk cohorts or outcomes? And the key is that the question should force replication and require donor aware inference. So once you have this question, decide the paper type early. That will give you a clear picture. Pick the product you are building, discover reanalysis, meta-analysis, integration, benchmarking, methods, atlas resources, or whatever you need, like for example clinical translation with a signature validated externally. This decision controls everything: how many data sets are needed, and what validations are required, and what figure sets will be credible additionally. So let us define the minimum package that can pass the review process. Well, the minimal package is at least two independent datasets, a reproducible biological effect that survives donor-aware modeling and sensitivity tests, a program or state or interaction supported by multiple lines of in silico evidence, and a shareable pipeline with a clean figure set. If any of these is missing, you can still publish, but usually you must reframe example method or resource, or you have to add some additional datasets. Next, where do you actually find the data sets that make this possible? Well, your go-to source are, like, there are many other database sources. Example, one of the very important one is Gene Expression Omnibus, which is freely available. Then SRA, then Array Express for raw matrices, metadata, broad single cell portal, or even SZ Select Gene for curated collections, Human Cell Atlas, large tissue atlases and figure sets, and Zenodo supplement. They all supplement linked from original papers. In practice, you will often combine one flagship data set plus at least one independent cohort for replication. But not all public data sets are usable. Here is how to filter fast these data sets. Well, the red flags that kill publication, well, too few donors or missing donors IDs, because you can't do donor aware inference, or no usable metadata for the comparison you need. Only processed embedding with raw counts, and the widespread ambient RNA, or even the low complexity with unclear handling and protocol chaos. This mixed and undocumented processing, they are also the killer for the publications. So if you ignore this, you will spend weeks and still fail review. So once you choose data sets, you must define outcomes and metadata precisely so that you can proceed further. And you have to define outcomes and metadata which you need. So start by registering your primary comparison, like case versus control, responder versus non-responder, time point, severity, all these factors you need to consider. Then list the batch variables like donor, library preparation, chemistry, center, sequencing run. Pre-specify the covariates like age, sex, smoking, medications, region, ischemic time. If you want translation, make sure outcomes exist. For example, survival, progression, response, all these matters. This prevents story drift later, and now I will show the end-to-end roadmap. This is the backbone of the whole lecture. Well, this roadmap is the full pipeline which is generally employed for the reviewers to examine your paper. For example, first of all, you have to acquire or harmonize the data. You have to do some quality control like doublet check, and also normalize, and you have to integrate the batches. You have to check all those clustering, or whether the annotations have been done properly or not. You have to check some statistics like differential expression, or even programs which are required for that. And then you have to also identify the state or trajectory or crosstalk between different cell types. You have to validate the package. The meta message is you have to decide the paper type early and plan replication from day one, because this is a very crucial step. 2 cohorts minimum, and bake validation including orthogonal evidence and robustness checks. If you follow this order, Your analysis reads like a method section reviewer trust. And let's start at the beginning. Well, the data download and the standardization. First of all, you have to prefer raw count matrices. There are many of them, like they have many different formats. For example, MTX, H5, CSV. The most common type one, these are the most common type, and the archive, the as related version, and the harmonized gene identifiers and mitochondrial naming conventions. These are all used, and they build a sample manifest mapping dataset followed by sample and donor collection, and then the conditions are checked, and then batch variables are checked, and the document every pre-processing choice. This sounds boring, but it is exactly what enables replication, debugging, and a clean supplement. So once the data are standardized, quality control is where you win or lose the credibility. So you must report per cell matrices. So there are many factors which you need to consider. For example, NUMI, then N genes, and percentage mitochondrial, often ribosomal too. And you have to show the distributions by sample and donor. So you should generally don't hide behind one global threshold, and flag the ambient RNA early. If you see broad low level of expression of markers across unrelated cells, you need to address it and do sensitivity checks. For example, you have to rerun the key findings under possible QC thresholds, and the goal is to show your biology is not quality control artifact. So that's very important. And next is the artifact that most often creates fake news statistics or doublets. So that you have to consider. So what are these doublets and multiplets? So these doublets creates hybrid clusters that look exciting but are fake. Use a standard tool like scrublet or doublet finder or scDBL finder. There are many more other algorithms which you can apply, and report the expected rate, and then confirm with diagnostics like high NUMI and mixed lineage markers. Then you have to recluster after removal and show your key signals persist. This is a classic reviewer request. If you do it upfront, you save the weeks later. With the quality control clean, we make the normalization choices explicitly. So next we will understand what are the normalization choices we have. So you have to pick one normalization strategy and justify it for your design. You have to do some log normalize, then scale or ST transform, or even size factor methods, and state how you select all these parameters, what you regress, like for example mitochondrial percent, cell cycle, if anything. The warning is don't over regress real biology. So before and after diagnostic for the key programs, so reviewers see you didn't have normalized away the signal. So that is also crucial here. So now integration and is necessary, so for the visualization and risk for the biology. So that's how we will move to the batch correction and integration. So this integration helps join the visualization but can erase real biological differences, and the options include anchors, harmony, scRNA, BBKNN, and the lateral models like SCVI, and your job is to evaluate, do batch mixes while no biological method or biological perception stays separated, and also you have to show a no integration baseline as an sanity check, and the reviewers trust you more when you demonstrate what integration changes. Then comes the clustering, which avoid pretending one resolution is the truth. In that case, we'll know that this cluster, like most of the cell types, they cluster across multiple resolutions and assess stability and interpretability. Use marker-based interpretation. Clusters should map to possible lineage or states. For example, many of the cells, they have their own particular cell-specific markers. One of the famous example, for example, you have some macrophage cells. So in that CD45 or CD11B, they should be like highly overexpressed, or they should be predominantly present compared to the other stromal cell types. Then guard against artifact clusters from, this is also a very common cause of artifact. So guard against artifact clusters from doublets like low quality cells or stress responses, and always provide an annotation table that is cluster to label to key markers. So that's how this annotation is where you make the atlas defensible. So these annotations, they combine manual curations, for example canonical markers, with reference mapping for robustness, and the automated label transfer is guidance, not truth. Because again, there are some cells which even though they explicitly show some of the markers highly overexpressed or predominantly they are present in that particular cell type, but still they might have some overlap to other cell types. So we have to be very very careful, and we have to do the back calculation and check whether they really those markers are present in that. If at all they are present, we have to check whether the other cell types or the other clusters also have or not, and we have to manually curate them. So explicitly flag ambiguous clusters and show uncertainty if needed, and include a dot plot or heat maps as evidence for labels. The theme is the annotation should read like an argument, not an assertion. You should not push it. You have to identify whether that cluster belongs to certain cell type or not. So now we reach the statistical core, that is differential expression done the right way. So in case of differential expression, you have to avoid naive per differential expression across donors. It inflates significance and will get flagged. Use pseudo bulk like donor plus donor and cell type, or donor aware mixed model with caveats, and report effect size and confidence intervals, not only p-value, and apply multiple testing correction. Then you have to validate the differential expression by complications what arises due to the replication across cohorts and alternative modeling choices. This slide is the difference between the pretty analysis and the publishable statistics. So that's what we need for the publication. And then we'll move to the genes, which are often noisy. The programs and pathways can be more publishable. So that's where we have to focus on. You have to convert your differential express list, because that's sometimes it's boring. So you have to convert them into a interpretable biology or enrichment graph and program scoring, and use curated genes like hallmark, reactome, cake pathway plus study specific modules, so program conservation across data sets and related cell types. You have to prioritize the programs with mechanistic plausibility and external support. Well, the point is the reviewers can argue about single genes. Conserved programs are harder to dismiss. That's how next we zoom into within cell type heterogeneity, where many stories stays. So the subcluster key lineage, for example, T cells, macrophage, fibroblasts, to identify states, define state markers and programs, then quantify state proportions per donor. So link those states to phenotypes, severity, response, time, all these are phenotypes, biological phenotypes. So you have to link to the time, region, outcomes, and most importantly, the reproducibility. Do the same states appear in an independent cohort with similar marker and direction of change? So when you compare proportions, the unit of the replication must be the donor. So we have to treat the donor as the unit of the replication for proportion comparison. Use the models appropriate for compositional data and include covariates, and report the uncertainty and effect size, not just p-value, and check whether the proportion shifts reflect biology or the quality control or the batch artifacts. This is another frequent reviewer trap. Don't let cell count differences overpower as a biology. If the biology supports continuous transition, you can add trajectory carefully. So now we will understand what is this trajectory and pseudo time is about, and how it guides your publication quality. So you have to use the pseudo time when you expect continuous transition, differentiation, activation, EMT like states like mesenchymal, epithelial to mesenchymal transition like states, one of the phenotype mostly prevalent in cancer, and quantify branch specific programs and link ordering to no markers, and avoid overinterpretation, because the pseudo time is not literal time unless independent supported, and cross check across tools and cohorts. Trajectory is persuasive only when it is consistent. So cell communication can be powerful, but it is easy to overclaim. These cell-cell communication tools, they generate many candidate interactions. So focus on a small, high confidence set, build an evidence chain. For example, you have to identify the ligand receptor expression, then followed by downstream targets, phenotype link. You have to find the replicate interactions across data sets and confirm directionality when possible. Present them as hypotheses supported by evidence, not definite causality. This language discipline keep reviewers on your side, and the major reviewer magnet is the orthogonal validation. Let's talk about that now. So the orthogonal evidence makes your story feel real even without a near benchmark. For example, single cell attack supports regulators and enhance link programs, and they also identify what is going on at the epigenetic level, and the spatial validates where states live and whether the interactions are possible. And bulk RNA-seq, they let signature generalize to large cohort at the tissue level, because you will have the bulk RNA, and link to outcomes protein level like site-seq or even proteomics, and they confirm the key markers beyond RNA. Choose one or two modalities that directly strengthen your main claim. And if you want a translational hook, a predictive signature is often the cleanest one. So you have to build a predictive signature after that. So derive a gene signature from a robust state or program, not a single data artifact, and test it in independent cohorts, often bulk RNA, and report generalization. Use appropriate evaluation and cross validation and calibration, confounded adjusted models, and interpret carefully. Then you have to identify those signatures which are either associated or not, unless mechanistically validated, and a clean signature figure can transform a nice reanalysis to a clinical relevant paper. Now we make reviews easy by proactively running robustness check. So for that, we have to check. Whether this reduced reviewer friction and replication is more than two data sets, and sensitivity to quality control thresholds and integration variants, clustering resolution, donor holdout or leave one study out checks, and negative controls. All these parameters are needed, like prematuration study, or which have the appropriate and also adjustable confounders, and also, for example, like age, sex, batch, chemistry, and if at all this metadata allow. This is how you show the signal is stable and not fragile. So if something breaks, it usually breaks in predictable ways. Let's name them. For example, one of the common failure mode in the paper is the typical one is the artifact clusters from doublet or ambient. Here you can see in the flow diagram that how the doublet, ambient, and stress, and the batch driven discoveries, the overclaiming function from expression and single cohort stories, and each has a fix. Remove artifacts and show diagnostics. Use donor-aware models and replications. Use cautionary language and triangulate evidence, and add an independent data or reframe as methods or resource. Next, we stitch everything into a clean narrative arc. Well, a paper reads well when the narrative is linear. Define the problem and the gap, assemble multicolor data and show quality control harmonization, discover a conserved state program and quantity with donor-aware stats, and propose a model supported by multiple analyses. For example, you need trajectory, crosstalk, regulation, and then validate externally, and release the reproducible code and discuss implications. You are not just showing results, you are building trust step by step. To make this tangible, here is a typical figure plan which you have to follow. For example, there are standard 6 to figures sets are required. For example, figure 1 will have like cohorts or quality control, integration, overview. Figure 2 will consist of like atlas, UMAP, as we discussed before. Figure 3 can have the lineage deep dive or the state of discovery and the markers which are used for annotation. Figure 4 can have like donor-aware differential expression programs and pathways, and so on. So this is basically your project plan. If you can draft these figures captions early, the analysis become much more easier to execute. So now let's talk about claims, how to write them safely. Well, prefer associated with, enriched in, consistent with, supports a model where, use we hypothesize when moving from expression to function. Avoid casual verbs like drives, causes, unless you have orthogonal experimental evidence. So you have to be careful when you are using the words while writing a paper by employing the single-cell RNA-seq dataset analysis from the public database. So be explicit about the limitations, observational data, heterogeneity data, residual confounding, all these you have to be careful about. This language prevents reviewers from forcing you into the impossible experiments, and the reproducibility is the prime factor, which is the last major pillar. Here is the checklist which you have to be careful about. You need a public repository with a pipeline, notebooks, figure regeneration scripts, pinned environments. For example, Conada, Docker, Ren, all these are there. You can go and check in Google. With fixed seeds, a data manifest with dataset ID, links, pre-processed nodes, and ideally a one command rerun workflow, like for example snake make next flow. This turns your paper into a reusable asset. Other people can also reuse your pipeline if they want to rerun, and that can enable the reproducibility, and people can reuse your set of pipeline, and the reviewers increasingly expect it. So if you are using human data, ethics and licensing matters. Briefly but explicitly, you have to be careful about the ethics and the IRB protocol and whatever is required as per your institutions, like ethics, license, and data use. So it's quite very important to understand that you have to verify the consent or the other terms and respect the access restrictions based on the consent form from the patients, and avoid exposing sensitive metadata. Be cautious with the small and subgroup reporting. Cite original data generators prominently and describe the data provenance clearly. Include a provenance table in supplements. You have to provide the details about the data sets, versions, processing, so your reuse is transparent and respectful. Finally, submission strategy, how to reduce surprise in the review process. So for that, you have to pick a target journal whose validation expectations match your package. Preprint early for feedback and priority. Anticipate reviewer asks doublets, pseudo bulk, batch diagnostic replication robustness, and have a response playbook with analysis ready to run. The best reviews happen when you already did what they were going to request. Let's close with a take-home checklist you can have the screenshot and use later. So your take-home checklist is are as written here. So the sharp question plus appropriate data sets with metadata like donor aware statistics with replication, as we discussed before. Validation via orthogonal modalities and or outcome generation, and robustness checks with clear figure and cautious claims. And you have to be mindful about the reproducibility, the code, environment, provenance package for reuse. If you can honestly tick all these boxes, you can publish without new bench work. Well, then in the next session, I will discuss about the hands-on training by using this particular paper. So if you have any question, you can direct to me. Thank you.